Hello everyone and welcome to today's The Scientist webinar. I'm Catherine Lloydell, Associate Science Editor for The Scientist, and I'll be moderating our discussion. Today, our speakers, Dr. James E. Crow Jr. and Dr. Joseph Jardine, will discuss potential neutralizing antibodies currently under investigation for SARS-CoV-2 treatment. We like our webinars to be interactive. We encourage you to send us your questions or comments at any point during the webinar, and the speakers will address these during the Q&A session following the presentations. To ask a question, simply click on the Q&A tab and type your query into the question box located on the right side of your screen. We'll try to address as many of these questions as we can during our Q&A session. The webinar will be archived on the Scientist website and we will send you the link via email within a couple of days. Please note that you will not be able to download the presentation slides. I'd like to take this opportunity to thank our webinar sponsors. Cynobiological. Cynobiological is an international reagent supplier and service provider. The company specializes in recombinant antigen production and antibody development. For more information, please visit www.cynobiological.com. Absolute Antibody. Absolute Antibody is a rapidly growing company with a vision to make recombinant antibody technology accessible to all. They offer antibody sequencing, engineering, and recombinant production as custom services, as well as a unique catalogue of recombinant antibodies engineered into new and useful formats. Thermo Fisher Scientific. At Thermo Fisher Scientific, our mission is to enable our customers to make the world healthier, cleaner, and safer. Thermo Fisher is at the heart of the global response to COVID-19, and we are working with government agencies and researchers to ensure priority access to instruments, consumables, safety supplies, and other products to address the outbreak. Find out more at thermofisher.com/coronavirus. Twist Bioscience. At Twist Bioscience, we work in a service of people who are changing the world for the better. In fields such as medicine, agriculture, industrial chemicals, and data storage, our unique silicon-based DNA synthesis platform provides precision at a scale that is otherwise unavailable to our customers. Acro Biosystems. Acro Biosystems is a leading manufacturer of recombinant proteins and other critical reagents to support the development of target therapeutics and diagnostic kits. To facilitate research on SARS-CoV-2 and help fight the COVID-19 pandemic, Acro Biosystems has developed more than 60 SARS-CoV-2 related products, about 2/3 of which are recombinant SARS-CoV-2 antigens with multiple tags and species, mostly expressed by HEC293, covering all the critical targets of SARS-CoV-2. 10x Genomics. 10x Genomics builds solutions to interrogate biological systems at a resolution and scale that matches the complexity of biology. Their rapidly expanding suite of products, which includes instruments, consumables, and software, have enabled customers to make fundamental discoveries across multiple research areas, including cancer, immunology, and neuroscience. BPS Bioscience. BPS Bioscience is a US-based original manufacturer with an innovative portfolio of over 4,000 products to assist in your drug discovery projects. As a primary manufacturer, BPS can supply products in large-scale bulk quantities and welcomes inquiries for contract and custom manufacturing within a wide variety of research areas. Our sponsors have provided us with some helpful resources related to today's webinar, and we have posted these in our handout section located on the right side of your screen. You can access and download these documents at any time during the webinar. And with that, let me introduce our first speaker, Dr. James Crow. Dr. James Crow is Director of the Vanderbilt Vaccine Center and the Anne Scott Carroll Professor of Pediatrics, Pathology, Microbiology, and Immunology at Vanderbilt. His laboratory studies the human immune response to infection for a wide variety of major human pathogens, including many emergent infections. He is the director of the Human Immunome Project, an ambitious effort to identify the sequence of all transcripts for human adaptive immune receptors. He is an elected member of the National Academy of Medicine, National Academy of Inventors, and other major professional organizations. His research group uses a very broad array of techniques, including molecular and cellular biology, state-of-the-art imaging, and flow cytometry, bioinformatics, and bioengineering approaches to attack scientific problems at the forefront of immunity research. The group has been recognized widely as a leader in antibody sciences, for instance, as the best academic research team, 11th Annual Vaccine Industry Excellence Awards, World Vaccine Congress, recipient of the 2019 Merck Future Insight Prize for Pandemic Preparedness, and large-scale research grants and contracts from the NIH and DoD, including the DARPA Pandemic Prevention Program, or P3. Okay, Dr. Crow, it looks like your slides are all up and running okay. So take it away. Thank you so much.
This has become our norm, and hope we can say some things that are interesting to you today. I'm going to be speaking specifically about human monoclonal antibodies for SARS-CoV-2 that we isolated in our consortium that's based at Vanderbilt Vaccine Center. So we were doing our normal work to make human monoclonal antibodies to various viruses, and we were about ready to launch a major effort on avian influenza as an effort we call a sprint, which is to go as fast as possible. And we were doing this on behalf of DARPA to do a capabilities demonstration. And we were going to launch at the end of January, so we were getting geared up to do that. And you'll recall January 19th, a man walked into an urgent care clinic near Seattle with the first case of SARS-CoV-2 in the United States. And we decided on the fly to terminate our efforts to launch a major antibody discovery sprint effort in the lab for an avian influenza, and we focused on corona immediately. And 7 days later, exactly a week from when this person walked in the door as the first case, I got cells at my home on a Sunday morning from FedEx. So FedEx is a Tennessee company, and we live in Tennessee. So the sample was flown on a special flight and delivery to Memphis, and this kind man drove 3 hours to give me the sample on a Sunday morning, and we launched our effort. It's a little bit terrifying because there were not any reagents, there was no virus, there were hardly any samples. It was one person in the United States. But we launched thinking that things would develop rapidly, and in fact they did. So almost immediately, Jason McClellan and others in the field gave structures of a receptor binding domain, NTD, spike trimers, um, EM, crystallography. There was just a plethora of information very rapidly. Scientists in China provided sequences of strains very rapidly. So this is the fastest we'd ever had access to the type of information during an epidemic that was necessary. And of course, Jason McClellan and others showed their dynamic features of the spike. So there's a closed spike and an open spike, and people showed very rapidly that the human ACE2 protein is a receptor, is used as a receptor and interacts directly with the receptor binding domain of the virus. All those residues were determined and available in the PDB. So um, because of the transition of these types of attachment and fusion proteins from pre-fusion to post-fusion states, McClellan and scientists at the VRC and others stabilized the protein with prolines, and we followed that lead and made reagents almost immediately. So we started developing strategies for finding human B cells secreting antibodies to SARS-CoV-2. But so essentially, we made many different antigens and workflows, but this is one I'll focus on. You start with streptavidin bead, and then you make recombinant antigen that's either full-length spike or the receptor binding domain or the NTD and various things like that. If they are biotinylated, they can be put onto streptavidin bead. We also were able to obtain recombinant receptor or human ACE2 protein with a tag, so we could really do a surrogate assay of neutralization, which I'll show you. So we had binding and neutralization reagents very rapidly, and so I'll show you some of the workflows that we did. We call these efforts sprints because we're going as fast as possible. Um, from when we start the effort, we want to be done. The first simulation we did last year was Zika as a model. Took us 78 days to sprint from sample to completing a full protection in primate study. So doing all the discovery, small animals, recombinant production or RNA production in primates. We did that 78 days last year, and we wanted to go faster this time because it was real and not a simulation. So in fact, we did two workflows, um, at the same time on the same samples. So we got blood we collected from many of the first cases in North America, in the United States and in Canada, working with collaborators, Helen Chu at Seattle, Mario Ostrowski in Toronto, and um, collecting even cases in Nashville where we live. So very early on, we got blood samples and isolated peripheral blood mononuclear cells, did stem cell based paramagnetic bead selection of B cells or B cell subsets or B memory cells. And then we did antigen specific sorting, typically by flow cytometry using a Sony cell sorter. And then this is where we split the workflow into two parallel pathways. On the left, um, we we put single B cells into a 10x genomics platform in which you can do single cell RNA seq and obtain antibody heavy chain and light chains back from single cells. And of course, they're paired because they have the same barcode from being in the same gem or um, vesicle, basically. And the other workflow, we took those antigen-specific B cells from flow cytometry and put them on a feeder layer that expressed CD40 ligand or CD154 and secreted IL-21 and secreted BAFF, and these are all factors that stimulate B cell expansion and differentiation. And so all of this stuff was done in a few days, and then downstream of that, the 10x genomics workflow, we do sequencing. We use Illumina NovaSeq typically, and sent those sequences to Twist Biosciences electronically, and they, using their in silico synthesis methods, made us thousands of antibody genes and cloned them on instrument by Gibson assembly into our expression vector, which we'd previously given them. And they ship us the full IgG expression vector as we want it. We take it right out of the box and transfect it into CHO cells. The other workflow, we took the stimulated cells and put them into a Berkeley Lights beacon device, and I'll show you more about how this works, but it's a single cell device that does high throughput assays on live cells. It was very nice to have live cells because then we could do binding and neutralization assays with single cells. But once we obtain cells of interest, those also can be used to make antibody genes, and those put into vectors into CHO cells. And then downstream of that, we made the recombinant antibody and did virus neutralization using wild-type virus initially, not pseudotype viruses, but eventually we compared with pseudotype viruses. So we were sending antibodies to our collaborator Michael Diamond at Washington University St. Louis, and he was using Washington 1 strain, so the virus isolated from the first case, and that was one of the cases we were studying to do neutralization. And we also um, did various binding assays. So we had multiple workflows ongoing, and I'll just show you the one on the right that I just described. So take B cells, which display the B cell receptor for the antibody they would secrete. So we take memory B cells, and they have a BCR on their surface. It's labeled with antigen, and that antigen is detected with a fluorophore. Then we did in vitro activation, as I mentioned, with CD40 ligand and cytokines and so on. And then we deposited those into individual wells on a beacon, Berkeley Lights beacon device. Um, and so just to show you what these assays look like, again, we have now our streptavidin bead with biotinylated antigen in gray. And then if a single B cell secretes what's shown as an antibody in blue here, and that binds the bead, then we introduce a conjugated antibody as an anti-IgG secondary. And so if the fluorophore labels the bead, we know that the antigen is now captured a human antibody, which is specific for SARS-CoV-2. And so these are bead-based immunoassays. And so up on the top right here, You can see it's dark, but these little green lines show you individual traps or pens, PEN, in which single B cells are deposited. And those B cells will fill those small volume pens within 20 minutes full of secreted antibody because they're stimulated cells. And out in the main channel are the antigen coated beads. And so as the antibody emerges from the pens, it's sort of like a volcano spewing lava out. The antibody comes out into the channel and binds to the beads. On the right, this is an actual photomicrograph of beads being labeled. Um, and this is the ectodomain of SARS-CoV-2 that was stabilized with two proteins put on a bead. So these wells have single B cells in them secreting um, ectodomain binding antibodies. We also could, in a different color, use receptor binding domain. That reagent we bought commercially was an FC Fusion with RBD. Um, and you could see in a different color beads um, with RBD on them lining up. So these are RBD specific S protein ectodomain reactive cells. So we've already even got some epitope mapping, um, and we got lots of these. And so just to show you what this looks like, these are hundreds of pens shown on this device. And this is about a 30-minute movie where out in the channel, you can see beads labeling for individual wells. I'll just play that again. It's pretty interesting to see the kinetics over 30 minutes um, with antibody coming out and labeling. So very rapid process, and then you can recover these cells. Um, so we did a lot of uh, that type of experiment, and um, so again with the beads, you identify the wells, and then we can recover single B cells using light. So the instrument uses light to move cells out and dump it into a tube. Now we also did essentially a single B cell neutralization test, which to my knowledge had never been done before, which is using a single B cell to secrete antibody that was then interrogated for neutralizing activity. And we did what was essentially a surrogate neutralization. So here is a light micrograph of 3 pins, and you can see there's B cells, individual B cells in each chamber. Now, this number 84 pen is shown now in the fluorescence view, and you can see it's secreting receptor binding domain antibody out into the channel. But we also did um, uh, a further assay in which we deposited receptor binding domain coated beads in the bottom of the well. And now the B cell is sitting right next to an NRBD coated well, and you can see that bead lights up in the bottom of the well. And if this B cell secretes antibody, it does not block and you still get labeling. But in another well, this 7574, it's secreting out into the channel, receptor binding domain antibody. It's binding the beads in the bottom, but if we look for ACE2 binding to RBD, this antibody blocks that. And so this is an antibody binding to the receptor binding domain and blocking that interaction with the receptor ACE2, which is a surrogate for neutralization. So we were doing binding and receptor blocking or neutralization type activity on single cells, and then we recovered those. And so that's the Berkeley Lights workflow. Um, and in parallel, the other workflow was PBMCs onto single cells on 10x genomics, sequencing those genes with NovaSeq, synthesizing with Twist, and then making thousands of antibodies synthetically. So we did both at exactly the same time with the same team. Um, the other thing that was needed for speed is a super fast and high throughput neutralization assay. And we, again, developed this on the fly. There was not a neutralization assay when we started, and we needed something fast. We didn't want to do plaque assays or focus reduction neutralization tests and the conventional things that are being done even now, and we're doing them now. We didn't need that for the discovery. The discovery had to be fast and ultra high throughput. So we used what's called cell impedance. So if you put cells into special plates, they will make an obstruction, which by making a monolayer, they create impedance across the electrical connections in wells. And this is done on a device called an Excelligence device, which is sold by ASEA. Um, and so it measures the impedance by monolayers. And over time, you can read these. They're in 384 well plates, and these are sensorgrams, and you can see the impedance. And so if you put the cells in over time, they make a monolayer and you get impedance. At the point where they're maximal or near maximal, we add virus, and the virus destroys the monolayer eventually, and you get cytopathic effect and you go back to no impedance. But if we put virus with antibody and the cell impedance is maintained, that means that the antibody has inhibited or neutralized the virus. So we did this with live wild type SARS-CoV-2 in a BSL-3 um, environment with Michael Diamond's group, where we basically worked this assay out and shipped them our instrument, which they put in the BSL-3, and did the assays. And also we did this in a somewhat sophisticated way in that um, we we were screening, so these were not highly purified, highly concentrated antibodies. These were supernatants, but we did 1 to 20, 1 to 100, 1 to 500, and 1 to 2500 dilutions of the supernatants. And you can see for this non-neutralizing antibody, we basically have a quadrant that's a dilution series. Impedance goes up and down, so it's non-neutralizing. But for this antibody in green, impedance goes up and up and up, even at 1 to 100, up and up and up. But if we dilute to 1 to 500, we lose the protection. So essentially, we were able to determine half maximal inhibitory concentrations, or IC50 values, in high throughput. And you can see we get the answer in 48 hours. So that was a 2-day neutralization test. By doing that, we have genes that are then in a mammalian expression vector, the heavy and light chain. We transfect those into CHO and make larger amounts, Chinese hamster ovary cells, and we can make purified antibodies. And here I'm just showing you photographs of real ELISA plates that we did in a more conventional way, where we were looking at ectodomain. Top right is receptor binding domain, bottom right is the NTD reagent, and the bottom left is SARS-1, SARS-CoV-1 full-length death. And you can see we got cross-reactivity for all of these antibodies. So very rapidly, we could figure out, are they RBD or NTD or S2 domain? Are they cross-reactive SARS-1? And then we were doing SARS-CoV-2 neutralization at WashU with Brett Case in the Diamond Lab, or with Ralph Barrick's lab. He has a nanoluciferase virus set up already, or wild-type virus. And we did neutralization in various cell lines and formats. These are potencies of some of the antibodies. You can see on the left, you have to be careful when you talk about potencies, because if you use wild-type SARS-CoV-2 virus, you'll get a certain set of numbers. So these two antibodies, 2196 in purple and 2130 in yellow, are 15 or 107 nanogram per ml IC50, which is quite potent. If you look at SARS-CoV-2 pseudoviruses, now they're down to 1 nanogram or below, and so pseudovirus overestimates the activity significantly. And a lot of the stuff you'll hear about or read about are pseudovirus neutralization, and we think probably wild-type virus neutralization is going to be more accurate in predicting, the physiologic environment inside a person. It's a picture of Brad and Swathi Srihari, and there are many others who were working the BSL-3, and it just makes the point that this is very laborious work to do live virus neutralizations with replicates and, all the work that had to be done, but they executed amazingly well, and we were able to determine IC50s for neutralization, also IC50s for human ACE2 blocking, which is a surrogate for neutralization, binding half maximal effective concentrations for ectodomain or receptor binding domain. And in purple are the most potent antibodies. And when you do that, you see the most potent ones really are ACE2 blocking. Um, which makes sense because they're blocking attachment and therefore they're very good at neutralizing. We did sequencing, of course, and analysis of all of the thousands of antibodies we were making. And these are just heat maps showing you heavy chain V gene on the y-axis and J gene on the X. And you can see by the heat maps for heavy chain, light chain lambda, light chain kappa. We got a wide diversity of antibodies, so we weren't getting the same antibody back over and over. There are some canonical antibodies that a lot of people seem to make, or public clonotypes that we have seen, and they match with genes that others have isolated and reported. But in fact, we get a wide diversity of antibodies. It's interesting when we look at the somatic mutation in terms of the percent identity to the germline, they're mostly 98% identical to germline. So these are not highly mutated antibodies that are hard to make like you would see in chronic HIV or diseases like that. These are easy to make, which is good because that means vaccines would probably be able to induce them in a lot of people. Then we wanted to know, now we've got thousands of antibodies, but where do they bind and can we group them into antibodies that bind to the same site, major antigenic sites. So this is a receptor binding domain in blue with a portion of that domain in orange, which is the portion of the RBD that's contacted by the receptor, which is humanase 2 shown in green. And so we used several antibodies, R2196, and then a previous antibody to SARS-1 from Crucell called 3022 as reference antibodies. And we started competing a lot of our most potent antibodies. And if they don't compete, you get 100% residual binding. But if they do compete, you see a much lower number. So for instance, all of these antibodies have very low numbers of residual binding because they're competed by 2196. And all of these are competed by 3022. So those are sort of overlapping competition groups. And then we had a third panel that did not compete with either one of those. So immediately we could see there are at least 3 antigenic sites that if you look on the right, but they all block ACE2 binding. So there's some subtleties in how you bind or recognize or interrogate the RBD. And we did point mutants to identify critical residues. So 2196, which is this top group, binds over here on the right to this purple residue, principally critical residue. And this bottom group binds to these yellow residues in RBD. So we knew they bound different places, and the competition suggested they could bind at the same time. So we took these two and studied them in terms of, could we generate a combination that would be helpful? And so this is single particle EM reconstruction on the left that Elad Benstein and our group did here at Vanderbilt, the EM core. And we saw simultaneous binding of 2196 and 2130 on one protomer of RBD in the open trimer form. And so it's clear you can get both of these antibodies on at the same time. And so next we asked, do they just add their activity, or is it possible that you would get some type of cooperativity or synergy? And we showed formally using a pretty complex synergistic neutralization algorithm. These are mathematical models and arguments that if you use 0 to 250 nanogram per ml of each of them and you mix them in various stoichiometries, you can see concentrations where, for instance, here at 6 nanogram per ml, there's really no neutralization. And at 63 of this antibody, there's no neutralization. But if you put them together, you have 96% neutralization. So at a concentration where neither is neutralizing, you get nearly 100% neutralization. And you can see the ratios are replicated on the right in a synergy map. So this was exciting because you in a combination, you have the potential to prevent escape mutations, which RNA viruses can do under monotherapy. But also we were getting more potency by synergy. We've tested monotherapy and combinations in, I think, 5 different animal models now. This particular model is a human ACE 2 transgenic model, which Michael Diamond introduced the human ACE2 gene into mice, which don't have the human ACE2 gene. He did it with an adenovirus using anti-interferon to accept that adenovirus, and then use wild-type SARS-CoV-2. And when we do that, um, the virus will um, make the animals lose weight if you have an isotype control, but our antibodies restore the weight curves. And if we look at the tissues, you can see on the bottom, the tissues are lacking inflammation, whereas isotype control has inflammation. So we've done this in adenovirus transduced animals, human ACE2 transgenics with Ralph Baruch, hamsters, um, and then finally we did non-human primates. So this is Dan Baruch's group um, at Beth Israel, did this as part of our DARPA group, and we did a prophylaxis study in which we gave antibody to monkeys, either the 2196 or another potent antibody, 2381, and challenged with wild-type virus, and then looked in the nasal swab or bronchial alveolar lavage. And on the right, you see the isotype control-treated animals have plenty of viral RNA, but we didn't see a single molecule of RNA in the nose or the bronchial alveolar lavage of either of these monotherapy treated groups. So this is very exciting because this enables development clinically, and each of these 2 antibodies are part of clinical development programs that'll be tested probably in August by 2 different companies, by AstraZeneca and ID Biologics. So just to recap what just happened, the sample that gave us all of the most potent antibodies arrived at our place in March 13th from our collaborator Mario Ostrowski in Toronto. We started the sprint. 25 days later, we transferred that to a pharma partner, and that antibody has now been manufactured GMP. It's going to be in clinical trials in August. So I don't know how you count days and speed, but in this particular case, we went from receiving the sample of blood in our hand to giving the pharma partner the neutralizing antibody sequences for CHO line development. So where we are now, we're doing more animal studies to figure out dosing and mechanism and distribution and that sort of thing. We're making structures by crystallography and EM. We're working with various manufacturing partners. We've already made GMP materials with them for phase 1 trials in Chinese hamster ovary cells. One, the partners made the antibody in plant cells and tobacco plants, actually. And another partner is developing mRNA for delivery of antibody genes. We've given these antibodies to numerous companies for incorporation into diagnostic kits. And then finally, vaccine companies are using these antibodies to characterize their vaccines or lot-to-lot variation in the antigenic sites because they want each vaccine lot to be reproducible. So in summary, we can move pretty fast to do this stuff, make antibodies that are very potent within weeks. And these approaches enable getting into the clinic very rapidly. And I think finally, it's fair to say that antibodies increasingly are going to be a major part of the future of preventing and treating infectious diseases. So vaccines are pretty complicated. They have complex safety profiles. They may or may not work in elderly, which is the highest risk population, whereas antibodies um, are generally have a very high safety profile. There's, closing in near 100 antibodies that are already licensed for the clinics. There's a lot of research and safety data on antibodies. And furthermore, they may work better and longer than vaccines. If you look in the primate study we did, the serum neutralizing titers in those animals were over 1 to 100,000. And these antibodies are all going to have 90-day half-life mutations. So we would expect at 90 days, a recipient getting that dose is still going to have a neutralization titer of 1 to 50,000, and you're never going to see that from a vaccine. And so the potency, the effectiveness, and the half-life of immunity mediated by antibodies could very well exceed the best vaccines that are going to come out, and they could be used in all people. So I think antibodies increasingly are the future. And so this is our team at Vanderbilt. Of course, this was pre-pandemic, so nobody had a mask on. And about a dozen of these people participated in the work because we, like you, were on lockdown during this whole time. And I mentioned most of the collaborators: Mike Diamond, Ralph Baruch, Dan Baruch, Unyun Lee at Emory is working with us. Helen Chu and Mario Strauss gave us samples. These are the companies that were supporting us with instruments and reagents. And I mentioned ID Biologics and AstraZeneca are doing the clinical trials. And we received support from Merck, DARPA, NIH. And being in Nashville, of course, we have people who are interested in what we're doing. And Dolly Parton gave money to Vanderbilt to support our work. And thank you for listening.
Okay, thank you so much for that presentation, Dr. Crow. As a reminder to our audience, you may ask questions at any time using the Q&A tab, and our panel will have a chance to address these questions after our next speaker. And that next speaker is Dr. Joseph Jardine, who kindly stepped in last minute after our original speaker, Dr. Sok, had to attend to a family matter, unfortunately. So thank you so much, Dr. Jardine. Dr. Joseph Jardine is the Director of Product Discovery and Optimization for AOVI based at Scripps Research. He is generally interested in the discovery and development of antibodies, nanobodies, and other protein-based therapeutics that can be used to combat infectious disease. His group uses a blend of synthetic and B-cell-based discovery approaches combined with structure-based directed evolution to engineer novel molecules, with a current focus on HIV, COVID, and snake envenomation. And Dr. Jardine, if you'd like to share your screen, we'll just check your screen is all up and running there for the audience.
All right, can you see that?
Going? Okay, great. And if you go full screen there. Fantastic. Okay, great. Thank you so much.
All right, yeah, thank you. Yeah, so as you said, this was a little bit last minute. So I stepped in and I managed to pull some slides together. And so this is going to be some slides that I put together paired with some of the stuff that Devin was going to talk about in this talk. And I'm going to try and change my presentation up a little bit as I go, so it's not as overlapping with Jim's. But you know, one of the things that was really striking about what's gone on the last sort of 6 months was a lot of scientists stepped in and tackled this problem, and we all came up with fairly similar solutions using fairly similar pathways and fairly similar timeframes. So I'm going to talk about what we did here. That was a collaboration between Scripps, IAVI, and UCSD. It was not as planned as Jim's. We are a group that spends a lot of time isolating antibodies, principally from HIV-infected donors. And so we've worked out a lot of those processes, but we weren't thinking about how to do this as quickly as possible. So when this outbreak happened, we sat down and we figured out how we could take the processes that we had in place and how we could rapidly adapt them to solve this problem really in an impressive amount of time. So just I have first here a slide showing all the people that were involved. So UCSD, we have, it was led by Tom Rogers, and he found the original cohort through the UCSD group. He came up with a live virus nude assay and then helped with the animal studies. We had Delhi, who came up with a pseudovirus assay, and that was really key in being able to do things quickly. Our group did the B-cell isolation, the antibody production, the validation. We had some grad students out of Dennis's group who did a huge amount of the hands-on work. We got protein from Ian Wilson, Rais Andrabi. Brian Briney helped with the sequencing, and John Taharo was one of the guys that helped set up the animal model. So very briefly, I mean, what we tried to do was model as best we could the HIV pipeline that we used. And so to find B cells from an infected donor, you really need three things. You need your cohort, and within your cohort, you need to have people that made a good antibody response. You need to have good access to the recombinant proteins. And that's one of the most important parts, because if you don't have good baits to find the antibodies, you're never going to get anywhere. And then lastly, you need some sort of viral assay to determine when you have a good antibody, because you'll find lots and lots of binding antibodies, but very few neutralizing antibodies. So in parallel, we started putting together all those three assays, and I'll talk about that later. And then once we had them going, we started rapidly screening the B cells in this, what turned out to be a 10-day pipeline that we put together. We could go from the fact sorting to getting the high throughput NEUT readout in really record time. And then from there, we take the positive hits that we got, we'd scale them, we'd characterize them more closely, because we did get false positives in there. And while all this was going on also, we were setting up an animal protection model so we could see if these animals protected the way that we hoped that they would. So just walking through this and trying not to have too much overlap with Jim, we were thinking about a lot of the same things that he was. So we knew that we wanted 2 baits to do our antigen selections with. We were using both the entire SARS-CoV-2 spike, and this protein became available really in record time. We got our first lot from Ian Wilson back, I believe, in February. And then we also, Jason McClellan sent us his plasmids, Rice was making some, and so we had good access to a lot of high quality spikes to look with. And then we also wanted to look with just the RBD. We were taking a sort of a page out of the HIV book in our logic here, where we knew that the RBD was how this protein would be attaching to the target cells. And so blocking that ACE2 epitope would be analogous to blocking the CD4 binding site with HIV. If you disrupt that initial attachment process, that's going to neutralize the virus. But one of the things that we also learned with HIV is that the most potently neutralizing epitopes are actually not CD4. They're epitopes on the HIV trimer. So one of them is going to be recognized by a class of antibodies, PGT121 being one of those that binds the N332 glycan in that area. And then the other one is going to be antibodies that recognize the trimer apex. So we knew that RBD would be a good target, but we didn't want to bias it against missing potentially very good antibodies that bound to the spike. So we spent some time and energy preparing both of these baits that were biotinylated for the single cell sorting. And we also were using them for the validation. So as the baits became available, we started screening our cohort to see who was making good antibody responses. And so initially we only had 17 people. We've got a lot more now. And they were not very far out from infection. At the very beginning, we did try and do some work with acute samples, and that didn't go anywhere useful. We got lots of binding antibodies, but nothing that neutralized. And so as the convalescent cohorts were becoming available, we realized we could utilize the same B cell selection technologies that we use for everything. And so that became our primary strategy. But what I've got here is just the ELISA binding to the SARS-1 and the SARS-2 spike from the 17 donors in our cohort. I've highlighted a couple that I'm going to talk about in more detail, but what we found from all of them is that we saw ELISA reactivity for nearly about 2/3 of our patients showed some binding, but there was a decent amount of variability there, and that was to the SARS-2 spike. We wanted to see if we could get cross-reactive antibodies because those would potentially be more interesting. So we were looking at SARS-1 as well, and that was a little bit more variable. In general, we saw at least a 10-fold reduction in titers compared to SARS-2, but there was a modest amount of cross-reactivity, so we thought that was kind of interesting. The story was different than when we looked at just RBD. So SARS-2 RBD, that had, again, pretty decent binding for a handful of our donors, but we saw almost nothing that cross-reacted with the SARS-1 RBD. So that said, we decided to not spend a lot of time looking for cross-reactive SARS-1, SARS-2 RBD clones, and decided instead to focus on the SARS-2 RBD because that's what's most relevant anyway. And so at this point, we had pretty good confidence that our probes were good. So in parallel, while this is going on, we were developing neutralization assays. And like Jim said, we wanted to have multiple assays because we didn't know what was going to be the best. So in both assays, we utilized a HeLa ACE2 cell line that we created, and that turned out to be better than the Vero E6 cells that we initially were using. So the first assay that we developed was an assay using live replicating virus from the Washington strain, and that was just a plaque-based assay. And that is probably the most realistic assay, but it's also quite slow and time-consuming. You have to sit there at a microscope and physically count the plaques. So in parallel, we also were creating a pseudovirus assay based on the same MLV that we use for HIV. And that would be detected with luciferase, and that's amenable to high throughput plate-based screeners. And so you could do much, you could sample a lot more with that assay compared to the live replicating virus assay. And we spent some time correlating the results of the two. We didn't see a huge discrepancy. In general, the pseudovirus is a little bit more sensitive, but if you look, we saw a pretty good correlation between pseudovirus and live virus, and these are with, this is patient serum right here. And we did see the same thing with the monoclonals later, but So overall, we did most of our screening with live virus, but anything that we found that were interesting, we tested with both. So going into the a little bit more detail about the patient cohort, we screened all of our patients for binding to the SARS-CoV-2 pseudovirus. We also made a SARS-CoV-1 pseudovirus. And the SARS-CoV-2 pseudovirus, we saw, again, a nice range of potencies. A handful didn't really show anything, and so we prioritized the donors that had the more potent neutralizing serums, because that's presumably where you'd find the better monoclonals. And against SARS-1, we really just saw nothing. So this correlated pretty well with the ELISA binding data with the RBD, not quite as well with the spike. So we didn't use a bunch of different selection strategies. This was really a brute force effort using the traditional antigen-specific B cell sorting. So what I've got up top here are just the standard FACS plots that we use. So you've got your scatter plots, you've got your live dead stain, and then we sorted B cells using CD19 and got rid of our monocytes, T cells, and everything else. And then when we first started, we were screening for IgG positive, IgD negative, but as more stuff was trickling out, we were continuously monitoring bioarchives because stuff was popping up in real time and it was changing. We did at one point try screening for just IgD negative, so we would also get IgA in the selected antibodies, and we didn't get a lot of interesting IgA hits, which we should probably go back and look at. But this was so initially we were just screening for IgG. And so then if you look down on the bottom left there, I've got a representative FACS plot from one of our selections where we've got, we did a dual staining with SARS-CoV-2 RBD and the CoV-2 trimeric S protein. So not surprisingly, you see very few cells that bind to anything. That's pretty standard. We saw almost nothing that bound to RBD alone. And that makes sense because the immune response was elicited against RBD in the context of spike. So turns out everything that did come through there was just noise. We saw a small number of B cells that did bind to both RBD and to S. That was, typically around 0.1%. And then we saw a couple percent that were S positive. And so on the lower right there, I've just got the different percentages for across all of the donors, again, highlighting the three of interest. And this generally matched what we were expecting from just given the size of the proteins. So then running through our really rapid expression pipeline, the first part really mimics what we're using for everything. So we would do our single cell, our antigen-specific B cell sorting, and that goes into 96-well plates, and you do your reverse transcription to make cDNA from your B cells. You then use a 2-round nested PCR where you amplify up the heavy chain and then do a second round where you add overhangs that will enable it to be rapidly cloned into the IgG expression vector. You run gels of everything that you get, and so you're going to have hits usually in about half of the wells. We had a little bit higher success rate than that, probably because we were sorting fresh samples. And then once we had, for all the hits that we had both a heavy chain and a light chain. We rearrayed them, cleaned them, and then did just the standard Gibson cloning reaction into our expression vector. So that pipeline or that workflow was pretty standard for what we normally do. Where we adopt things to be really high throughput is rather than transforming the Gibson reaction and going down to single colonies and picking a colony and sequence confirming that it was what we thought it was, we spent some time optimizing the efficiency of our Gibson reaction so that we could get almost 100% clone. You'd sequence 5 colonies and you'd have 5 in-frame sequences. And so then rather than going down and sequence confirming, we would just transform into bacteria and then outgrow the entire bacterial culture in antibiotics, prep the entire culture. And so you're going to have, predominantly properly cloned antibody, but there probably is a little bit of closed vector in there. And then we would transfect that in 24-well plates using XB293 cells. So all of that could be done in really about 4 days. And on day 4, we would put them in culture, and then you have to spend another 4 days or so letting the antibody express. So on about day 8, we would end up harvesting the supernatants. And then rather than purifying, we just went straight ahead and used that for high throughput screens to look for, first we looked for the presence of IgG to see whether or not it expressed. We then looked to see whether the IgG bound to SARS-1 or SARS-2, S or RBD. And then we also looked to see whether or not it neutralized in a high throughput pseudovirus assay. And so this slide is a little bit busy. We're trying to put all of the data together into one overall slide. So we sorted about 3,000 total B cells, of which a little over 2,000 we successfully had a heavy chain and a light chain that we cloned and went ahead with transfecting. So what I've got here, the scale here in dark blue is going to be either high levels of binding or neutralization, and then in white is going to be low binding or no neutralization. And so if you look at the number of antibodies or the number of wells that expressed in IgG, we really were successful with about 90% of them. So this high throughput workflow had a pretty good overall success rate at just antibody expression. Of the antibodies that expressed, about half were reactive to SARS-CoV-2 S protein by ELISA. And this, again, is pretty consistent with what we normally find with our workflows, where normally about half of the antibodies that come down are just non-specifically, they're just noise that were selected, but half of them are antigen specific. And then of the S reactive clones, 15 or 20% of them also bound to RBD. So you can see it's a much smaller subset that are RBD reactive. And then we have BG505 trimer in there just as a control to get rid of the sticky clones. So there was a couple of sticky clones in there as well. So this data really nicely tracks with what we saw from the frequency of binding during FACS. What was interesting is when we looked at the neutralization, we just did a threefold dilution series where we looked at just the undiluted supernatants from the XB transfections. We looked at 3x diluted and 9x diluted, and so it turns out that the undiluted really did have a lot of background. So most of that we sort of started ignoring, but we noticed there were a modest number of hits in the threefold and ninefold diluted, and if you look at them, they almost all track back to the antibodies that react against the CoV-2 RBD. So we saw a couple. I've got that highlighted. Sorry, up here I've highlighted the ones that we actually went ahead and made. So you can see most of the antibodies are over here next to the RBD. We had a couple of hits that did not bind to the RBD, but really it was pretty clear from this that the good neutralizing antibodies were going to be RBD reactive. So the ones that showed good hits, we went back and actually sequenced those clones so we knew what they were, and we produced them at scale for careful characterization. So what I've got here is I've got the neutralization curves from a handful of these antibodies, and they really were kind of all over the place. So we had some that didn't neutralize at all. They were just false positives from our high throughput screen. We had a modest number of antibodies that showed kind of low microgram per mil IC50s, but we had a handful that were quite potent. So we had some that were neutralizing in the low single digit nanogram per mil. We now have a couple that are actually better than what I've got on here. So overall, we were pretty happy with the efficiency of isolating these. And we really, I think at this point, it was about 15 days from when we started that we had this data. So it moved incredibly, incredibly quickly. So the next thing that we wanted to know was, we'd already worked out that most of the neutralizing antibodies bound to RBD, but we really wanted to see where these things were binding. So we did some epitope binning. And what we found is our RBD antibodies really fell into to two different epitopes. So we call them RBD-A and RBD-B. There was also an RBD-C epitope that binds CR302 that we didn't find any antibodies we didn't find any neutralizing antibodies that reacted against that. But the majority of our neutralizing antibodies were in RBD-A. And then we also found a couple of different epitopes on the spike that we were pulling down antibodies that were giving hits in the neutralization assay. So if you go over and you actually now compare the epitopes to the IC50s that we measured, it was quite startling. All of our good neutralizing antibodies really were RBD-A. We found a single one that neutralized well on RBD-B, and the spike reactive clones really didn't do much of anything. So from this, we hypothesized pretty quickly that probably what we had were antibodies that were directly blocking the ACE2 interaction, but wanted to confirm that. So we went on and we ran a competition assay. And the way this was set up was we had our cell line that was overexpressing the human ACE2, and then we had our biotinylated RBD or trimer that we would oligomerize on a fluorescent streptavidin. And so in the absence of an antibody, when you mix the fluorescently labeled RBD or trimer with your ACE2 cell line, you would see nice fluorescence. And then we could measure the amount of competition by mixing in the antibody. And so this not only looked to see did it compete with the epitope, but it actually took into consideration a little bit of the antibody binding kinetics. And so you can have these antibodies that partially compete because maybe they have low affinity, and so they disrupt the overall interaction, but at some point they get a little bit competed off by the cell. So we ended up with this really nice neutralization versus competition plot here. And this is not what you typically see when you look at data. So we were quite pleased to see this. And we were also pretty confident at this point that our RBDA epitope was the ACE2 binding site, and these antibodies were directly blocking. So a couple weeks later, we actually were able to confirm, a couple weeks after our paper was published, we were able to confirm that when Ian Wilson rapidly solved the structure of one of the antibodies bound to the RBD. So I've got that over here on the right. We've got CC12.1, and that's bound to the SARS2 RBD. And if you superimpose that with the ACE2 structure that's previously been solved, you can see it's not a minor clash. It ended up being almost a complete overlap of the epitope, where the antibodies is, the majority of it is now engulfed in the surface of ACE2. So this is how you would really design something to clash if you were trying to do it rationally. We found a lot of those antibodies. And then one of the other things that was a little bit striking about what we found is we had almost no antibodies that cross-reacted and were able to neutralize SARS-1. In our paper, we have a single antibody that does that. And what it turns out that does make pretty good biological sense, because the ACE2 epitope is it's not very conserved between the two different viruses. So what I've got here is I've got a crystal structure of the ACE2 bound, and that's in dark blue, bound to RBD in gray. And I've highlighted in orange all of the mutations between SARS-1 and SARS-2. So overall, there's not a lot of conservation between these two in the epitope that our antibodies were hitting. And if you look at the CR3022 or what we guess is the RBDB, there's much more conservation. So that makes sense as to why we really had COVID reactive antibodies that didn't cross-react with the earlier strain. So all of this was being done, as quickly as we possibly could. And in parallel, Tom and John were working on an animal model so that we could test prophylactically whether these antibodies would be protective. And so we ended up using the Syrian hamster, and that was based on previous work that had been done. So this hamster has an ACE2 that enabled it to be infected, and we did a little bit of work to figure out how to dose them. But overall, it was pretty straightforward getting the model up and running. And so we wanted to look with two different antibodies and see whether or not either of them could be protective. So over there on the left, I've got CC12.1, which is one of our more potently neutralizing antibodies. It's about, I think it's 15 nanogram per mil IC50 against the pseudovirus, and a little bit more against the live virus. It was 20 or 25 maybe. And then we also were using, and CC12.1 recognizes the RBDA epitope. We also wanted to look at one of the spike binders we had that showed a little bit of neutralization. And so we used CC12.23, which is a couple logs lower IC50, and you don't even get to completion in this assay. But it did show some neutralizing potential. And so we wanted to see, are these able to protect our animals from COVID challenge? So we set up a couple of groups. We had 6 animals per group. The antibody was delivered by IP, and then the next day we did our intranasal challenge with, I think we used 10 to the 6 virus. We gave the animals 5 days, weighing them each day as a readout of protection, and then at the end of it, we sacrificed them and we took out their lungs to look at the amount of virus that was there. And since we also didn't know what level of antibody was going to be protective, we went ahead and did 5 different doses. So first we had a DEN3 isotype antibody control that does not neutralize SARS-2. And we had 5 doses of animal receiving 2 mg per animal all the way down to 8 micrograms. And that translates pretty nicely to what you could do in a human. So 2 mgs is about 15 mgs per kg in a hamster. And that's something that's readily achievable in a person. And so initially this was done, it was done blinded, but it was pretty apparent that there was major differences very early on. So at the end of our 5-day experiment, the control animals had dropped about 14% of their body weight. But one of the other groups, which ended up being our high dose group, the animals actually gained a little bit of weight because they're given unfettered access to food and they keep eating and they keep growing like they normally would. So we saw complete protection at the 2 mg dose and the 0.5 mg dose. We saw some protection at the 125 microgram dose, and then we saw no protection at all at 31 and 8. What was a little bit scary was we actually saw maybe some evidence of enhanced disease where the animals lost a little bit more weight compared to the control group. So we are actively following up on that to try and understand what was going on there. When we looked at the lungs, not surprisingly, the animals that had the most virus in their lungs were the ones that had the DEN3 control antibody. We saw really no antibody in the 2 mg all the way down to 125 microgram groups. This is going to be at the level that your sort of viral debris is going to be giving you a signal at. And then we saw some virus in the 31 and the 8, although it was less than the control group. And that's evidence that it did to a degree control the replication of the virus as you'd expect with a low level of antibody, but it wasn't enough to protect. So CC 12.3 was this is really what you'd like. I'm sorry, 12.1 was really what you would like to see in protection, where you've got this nice dose dependent effect, and really at doses that should be achievable in people, you saw complete protection. So in contrast to that, the 12.23 showed no protection at all in any group. And so I've just got the summary of we actually let this one go out to 6 days, weighing them on day 3, 4, and 6. And there's no difference in weights between the high dose and the control. So really everything was within the air. And so not surprisingly, if you have an antibody that doesn't neutralize very potently, you don't see protection. So this fit quite well with our model. So just going to summarize what we ended up finding, we screened 17 individuals and produced over 2,000 antibodies. We found antibodies against 6 different epitopes. Not surprisingly, the antibodies that directly compete with ACE2 are the most potent, that fit with the literature. We were a little bit surprised that we found antibodies that bound with high affinity to the spike that didn't neutralize at all. So we've now found a lot of these and don't really understand what's going on there. We have good lead candidates to move forward, and we also established that the Syrian hamster model works well for protection of disease. So that was our high throughput antibody optimization effort. It took us about 8 weeks to go from beginning the project to submitting the paper, which is, and that workflow would normally take 2 years. So there was a lot of hard work by a lot of people, and not a lot of sleep. So it was quite impressive what they all did. So now that we've got some good lead candidates, we're looking to see what we can do to make them better, because you want an antibody that neutralizes as potently as possible. We're thinking a lot about how you go about delivering these. And one of the things that we've been working on a lot is improving the potency of some of our HIV antibodies through affinity maturation. So what I've got here, I've got 3 different HIV antibodies. On the left is an antibody called PGT121, and that recognizes that N332 glycan I mentioned earlier. And then each one of those dots is the IC50 against a pseudovirus. And so it neutralizes a lot of viruses quite potently. And then up at the top, there's a handful of viruses that it doesn't neutralize well either. And this is true of most HIV antibodies. You have a lot of diversity, and so there's always some that are not neutralized. But we put PGT121 through our affinity maturation protocol, and we've got variants of it now that bind with much, much higher affinity, and that overall is translated to substantial increases in neutralization potency. And so this is one of our priorities right now, is to see if we can apply this to SARS. And just to show a little bit of that data, we observed, the thing that actually made us think that this was going to work well against COVID as well, is we looked at the IC50 of our RBD-A reactive clones, and that IC50 correlates quite well with the KD. So this is not quite as good as our competition correlation, but you can see a pretty high correlation between IC50 and KD. And so we reasoned that if you can further improve the IC50, further improve the KD of these antibodies, you should see at least some gains in neutralization potency. And so I have some of the early data right now. We're running it on all of our antibodies, but one of the first things we wanted to try this strategy on was actually CR3022. And so historically, this is one of the first antibodies that people were looking at. It was isolated, neutralizes SARS-1. And Ian Wilson solved the structure of it quite early on in this whole process, where it's bound to RBD. So we knew that it bound. A lot of groups had reported that, but it didn't bind with high affinity. And so, we had speculated that could have been why it didn't neutralize. There was also a lot of hypothesis that it could be a steric occlusion of RBD on SARS-2, but we thought, this could very well be a kinetic approach. And so over on the right, I actually have the SPR trace of the CR3022 binding to the original SARS-1 RBD, and you get your sub-nanomolar binding. And then binding to SARS-2 RBD, you end up with 25 nanomolar binding, but this very, very fast off rate. So this looked like a problem, and you typically don't see this with neutralizing antibodies, and that was something that we wanted to try and fix. So I emailed one of my friends over at Twist and got him to make some libraries, and then we put that through our standard affinity maturation process, where basically what we do is we take our starting antibody, we make yeast display libraries of that antibody where you have lots and lots of variants of it. So I split things initially into a heavy chain and a light chain. So we screened 5 million different heavy chains, 5 million different light chains, and then eventually put them together to screen the best of the best. The antibodies displayed on the surface of yeast, and we use FACS to isolate the antibodies that bind with the highest affinity. We then express a number of variants of those and then go on and characterize them to see what our modifications really do. So we're running through this pipeline in parallel to our antibody discovery process with CR3022. And it ended up working quite well. So over here, I've got the results from our first round of optimization. And so on the left, I've got ELISA. So up top, I've got the binding to the SARS-1 trimeric spike, and the wild type antibodies in black. So that shows pretty high affinity. And all of our design variants are in gray, with 3 that I pulled out in different colors. And we can see slightly increased affinity by ELISA, but really they bind pretty comparably to the SARS-1 spike. But in contrast, SARS-2, we saw substantially enhanced ELISA binding. So the wild type CR3022 shows pretty poor binding and plateaus out quite early. But all of our design variants look pretty much like they do binding, or the original antibody binding to the SARS-1 spike. It was a little bit more apparent when we looked at the SPR sensograms. So over there, over on the right side, I've got the binding to SARS-1 RBD and the binding to SARS-2 RBD. So again, you've got your original CR3022, where you've got high affinity SARS-1 binding, and you've got this very, very fast off rate to SARS-2 that looks almost small molecule-like. But all of our design variants had substantially increased affinity. So we got about 1,000-fold potency boost here. So we were really quite happy with this. We went and we tested them against neutralization. We actually were able to rescue neutralization of CR3022 for neutralizing the SARS-CoV-2. So I just have the Newt assay over here on the left for for neutralizing CoV-1. We got a modest increase in IC50. But CoV-2, we have, for most of them, we've got about a microgram per mil, maybe a little bit more potent neutralizing, compared to the original CR3022 in black that does almost nothing. So we were really happy with this, and are now running the same optimization program on all of our lead candidates to see if we can get a 5 or so fold bumped in the IC50. And so then just to close out, Jim did a fantastic job finding these antibodies incredibly quickly. I think he actually was the first person to get this done, and he got them out to companies as rapidly as possible. One of the things that Avi has been stressing is we're looking to not just get the antibodies out to the United States and Europe, but we're really looking to see how do we make them available in low and middle income countries as quickly as possible, because there typically tends to be a substantial lag time there. And so just like he discussed, I don't need to go too far into detail here, but we are looking at both therapeutic indications as well as prophylactic indications. Therapy has obvious benefits, but as he talked about at the end, with antibody prophylaxis, I think there is growing evidence that this is going to be a field that people are utilizing more. And RSV has demonstrated that this can work quite well with palivizumab. And if you look at the people that are most susceptible to SARS, a lot of them are going to be people that don't respond terribly well to vaccines. They're going to be the older population. So we're looking at these two indications, and then really we're looking to see how do we make this affordable, because in developed countries you can spend substantially more money on healthcare, but if you're trying to make this accessible globally, you have to really factor in your cost of antibody production. And so scientifically, we're trying to improve the potency of the antibodies so you can give lower doses. And, we're following the same overall workflow for the PGT that I showed you, where if you can get a 50-fold gain, we're hoping that you can give a substantially lower dose and achieve the same level of protection. And then we're also working with a number of manufacturers to see how can we produce the antibody as cheaply as possible. And so at this point, we're pretty confident that we might be able to get the manufacturing down to really about $20 per gram. And that's getting to the realm that this is something that could be affordable. So just the final workflow, and these are Devin's slides, and it's what he's been very passionately working on. We've got our lead candidate. We're using every bit of innovation we can as far as half-life extension, affinity optimization to try and improve the potency so that we can minimize the amount of dose that we need, and ideally be able to deliver it subcutaneously, because if you have to go to a hospital for an IV drip, that's going to be problematic. And also anything we can do to improve the overall yields of our antibody. We're also looking at all of the partnerships to produce these antibodies. And so we're looking not only, again, for getting it to the US and Europe, but also how do we get this to other areas of the world that really need it. While we're thinking about the global trials, we're making sure that it will be approved in as many places as possible. So a lot of countries will allow drugs that are FDA approved, but not all. And so if they don't recognize FDA approved products, we need to make sure that we do the trials in that country as well. And then lastly, as we're doing all of this, we need to make sure that other countries are prepared to get access to this antibody for the people that need it. So this is one of the major efforts right now going on at IAVI. So it's not just going to the United States and Europe, it's really trying to figure out how do we use this in a global setting. And COVID is providing an interesting opportunity for that, because for a long time it was thought that antibodies were not going to be affordable outside of the developed world. And if we can make this model work for the low and middle income countries for COVID, we should be able to do it for other things such as HIV. So overall, this was a huge collaborative effort where everybody here really dropped what they were working on and all started working, just on this. I think most of us were doing 7 days a week, so the long 10 to 12 hour days. So it was it was a pretty ridiculous sprint, but we did get there in the end. So thank you.
Okay, thank you so much, Dr. Jardine, for another fantastic presentation. The audience is loving both of your presentations, by the way, just to pass on some kudos there. And also they want to congratulate you both on, such speedy work. So the audience has already submitted several questions, so we'll get straight to them. So there's been a few questions actually about antibody-dependent viral enhancement. And perhaps Dr. Crow, you could go for this first. Does having neutralizing capacity mean that the antibody will not mediate antibody-dependent viral enhancement?
This is a good question. So antibody-dependent enhancement is a concept that has grown mostly out of the flavivirus world. So antibodies to the fusion loop on flavivirus envelope proteins, at least in vitro, will, if they're non-neutralizing, and most dengue or other flavivirus fusion loop antibodies are not very good neutralizing antibodies, they tag the virus, they put an FC onto the virus, the FC binds to an FC receptor and takes that virion particle, which is not neutralized, into something like a macrophage, which is permissive for replication, and you get more dengue virus out in vitro than in the absence of the antibody. So that's the predominant model for ADE is dengue and flaviviruses. There are some specific animal experiments that have been done with other coronaviruses in which people have observed antibody-dependent enhancement in vivo in rare situations. So the question was raised, does it occur in SARS-CoV-2? And I would say most experts do not find evidence for a medically relevant ADE being observed in nature by people that are previously exposed and re-exposed, or previously infected and re-exposed, has not been seen, to my knowledge, in experimental immunizations, of which there have been a lot of people who were subsequently exposed. And in most experimental situations, it doesn't happen. So I think the general consensus is it's not a common or major factor in SARS-CoV-2. Now, nevertheless, I mentioned that some instances of coronavirus ADE had been shown in certain animal models. So there are ways to look for ADE in terms of in vitro assays, and certainly many of us are sort of looking at that. There are bead-based assays that measure binding to FCRs or ADCC activity or ADE activity, and lots of us are doing that sort of thing. An alternative would be to knock out binding to FC receptor. And at least one of our partners has chosen that path, and so they've introduced mutations into the FC region of the recombinant antibodies that are being used in the clinic that do not engage FCR, so they would not have the theoretical potential to do that. I would say most groups that are testing antibodies clinically are not doing that, because you're likely to lose some ADCC when you do that, which might be beneficial. And again, most people don't think ADE is common or important, but at least one of our partners has fully knocked out FCR engagement. So I think it remains to be seen whether it occurs or is it common fully as more epidemiology is done. But that's where we are, I think.
Okay, thank you for that, Dr. Crow. Dr. Jardine, perhaps you may be able to answer this one. Emerging evidence shows that natural antibody levels are falling in most of infected people after just a few months. What are your thoughts on this, and how does that relate to manufactured antibodies?
Yeah, so I think there is some good evidence of that. It obviously takes a while to really work that out, because you've got to wait some time to see what antibody titers do. But it does look like the titers from natural infection do fall off fairly rapidly. Whether or not we see the same thing from vaccination is an open question. I think that is the billion-dollar question right there, because the natural infection via respiratory virus is going to be different than what you're getting from a vaccine that's delivered. Particularly a vaccine that's delivered with some mRNA technology or something like that, that's going to be an overall different immune response. I think that the space for the antibodies really is complementary to a vaccine, because we do know that they will fall off. I mean, this is very well worked out technology. Even the best behaved antibodies have a very predictable half-life. You can certainly make it worse by having a sticky antibody, but you can take your perfect antibody, throw an LS extension on there, and you'll probably still need a shot every sort of 3 to 6 months. So I don't think that one is going to replace the other, but we are actively watching this field where the vaccine titers, or the titers from natural infection do appear to fall down pretty rapidly, and it's concerning. It's not what you'd like, but at the end of the day, we do have to wait and see what happens over time, because this is it's a slow process.
Okay, thank you for that. The next question is about, so did you look, this is for Dr. Groh, sorry. Did you look at the lung of the animals and whether it's protected by antibodies? And did you look at other organs as well? So for example, virus has been found in the brain even.
Well, we've used 4 or 5 different animal models. I can say we have done histopathology in the various mouse models, and we see an improvement in histopathology correlating with the lack of virus replication. We are in the process of doing the non-human primate histopathologies. When you're using RNA readouts or even plaque assays as your readout for replication, you can get data in 1 to 3 days, but the processing of tissue and staining and evaluating and scoring them and all actually takes several weeks. So we've already published our viremia or viral load data from lung and primates, but we're still working through the histopathology there. But I think, you know, the downstream thought process, maybe what you're getting at, I think the sooner you add antibodies into an exposed or infected person's body, the better they're going to be in terms of interrupting disease. So they're almost always likely, antibodies of these regimes of potency that we're talking about today, they're always going to have an antiviral effect and reduce virus. But if you give them before infection or exposure, they're going to work great probably. If you do post-exposure prophylaxis before symptoms, they're still probably going to work great. If you are treating people with mild upper respiratory symptoms, I would hypothesize they would probably prevent severe disease from occurring or hospitalization. But once you start getting into hospitalized patients with severe disease, hospitalized patients on a ventilator, then simply having an antiviral effect may not be sufficient to reverse disease because now you already have an inflammatory cascade ongoing. So one thing you might see in future is a convergence of clinical trials that are using things like anti-IL-6 or even dexamethasone is being used clinically sometimes. So you have anti-inflammatories. If those are paired with antiviral, that might be better in the setting of severe ongoing disease with established inflammation than an antiviral antibody alone. So sort of depends on what population you're talking about and what's the goal. If the goal is to have an antiviral effect, these are going to work, but they're not likely to reverse established histopathology, I don't think.
Okay, thank you, Dr. Crow. Dr. Jardine, this question is for you. Are nanobodies preferable to full antibodies from maybe an immune perspective in avoiding unwanted immune effects?
Probably not, no. So nanobodies are, they've got some interesting applications. They tend to be, so an antibody, just to back up, it's going to be just the VH portion of an antibody. And so you can either have that just on itself, so it's going to be 20 some kilodaltons, or you can fuse that onto an FC domain, and so you'll have all of the effector functions and the half-life extensions. It is quite artificial. So we've got some humanized versions that we're playing with right now, but if you look at most of the nanobodies that are out there right now, they came out of a llama or an alpaca or something, and so they have some advantages and disadvantages. One of the advantages that's quite interesting about them is they tend to be really, really stable. So that opens up all kinds of interesting possibilities where if you wanted to try and nebulize it so you could deliver it directly into the lungs, you could do something like that. We're looking at antibodies right now for snake envenomation because they may not need a cold chain, and so that could be huge when you're trying to disseminate these things around parts of the world that don't have cold chains. But as far as, and I guess one of the other interesting things that has come out with nanobodies and HIV is that they bind with an overall different approach compared to a human antibody that has a heavy chain and a light chain and has these 6 bulky loops. So they can access the virus in ways that you wouldn't normally see, that doesn't have a lot of selective pressure to avoid. In the context of SARS, I don't know that there's any real advantage to it aside from the nebulization. I would guess if you administered both a recombinantly produced antibody that you isolated from a patient versus a nanobody, you're going to see the immune response against the nanobody, much more often than the antibody. So there's pros and cons to them. You can do some interesting stuff with them. It also becomes you can do some interesting bispecific type generation as well, where you fuse nanobodies onto your antibody, and so now you can have multiple points of contact with the same molecule, and you don't have to worry about heavy light pairing that sort of complicate bispecific generation. So we're kind of looking at a number of things here. Our library was available. We decided to start screening it anyway, so we'll see what comes out of that. But our primary interest right now is, in fact, antibodies. The nanobodies is a fun side project.
Okay, thank you for that. Dr. Crow, could you comment on the safety of neutralizing antibodies in immunocompromised people?
Well, yes, this is one of the major advantages. So in immunocompromised populations, vaccines are not likely to be particularly relevant or effective. But typically, you can give monoclonal antibodies or immune globulin of various types, polyclonal immune globulin, to immunocompromised patients. And this is done routinely in hospitals. So many cancer patients are given various types of immunotherapy with monoclonals. Even to this day, anti-thymocyte globulin, which is made in horses, I believe, is sometimes given to stem cell transplants. So the safety profile of antibodies in immunocompromised people is very, very high, and that's one of their advantages. I mean, there's some particular cases. If you're IgA deficient and you get IVIG, you can have kind of an anaphylaxis sort of response. So there's particular types of immune derangement, that might set you up for adverse reactions. But in general, passive administration of antibodies to immunocompromised patients is very safe.
Okay, thank you for that. And we have time just for one more question. Dr. Jardine, you mentioned there's little cross-reactivity with SARS-1 with one of the antibodies you were looking at. Would there be any cross-reactivity with other coronaviruses, such as the 4 that cause the common cold?
Yeah, so we're looking at that right now. We don't have any data on that yet. We think all of the antibodies that we isolated, we think almost all the antibodies we isolated were probably primed by this immune response, because they were really not very mutated at all. It was kind of shocking. I mean, I spent my graduate years working on an HIV antibody called VRC01 that's 30% mutated from germline, and what we typically saw was much like what Jim saw, where you get really 1 or 2% mutation, and that's enough to get a neutralizing antibody. The SARS-1 cross-reactive antibody, we've got a pretty good idea of where that binds, and that's just going to be an area that's conserved SARS-1, SARS-2. That area certainly doesn't appear to be, at least from a sequence point of view, conserved in MERS at all. And I don't think it's conserved in at least a couple of the other coronaviruses that I've looked at. But that's, I think Rais is right now testing to see which of these antibodies cross-reacts. He's really interested in the cross-reactivity to the common coronavirus strains. So that wasn't really something that we saw a lot of. I think probably because we screened for the neutralization really as the readout. I would be surprised if we went back and looked through all 2,000 antibodies if we didn't have a lot more cross-reactive ones. If you look at a lot of the antibodies that were isolated by Adimab, for example, they screened somebody that was initially infected with SARS-1, and they screened for SARS-2 cross-reactive antibodies. So really everything they had was cross-reactive. So I think it's really just how you what your source is and how you do your screening. But since we were looking for really the most potently neutralizing antibodies, those are going to be the ones that specifically hit the ACE2 epitope on RBD, and that just happens to be really different on this particular coronavirus. And so I think that they're there. We just need to, if we really wanted them, we'd have to go back and look for them more.
Okay, thank you so much. Unfortunately, that's all we have time for today. If you have any further questions, please consider reaching out to the speakers directly. Their emails are shown on the screen right now. As a reminder, the webinar will be archived on the scientist website, and you'll receive an email notifying you when the on-demand webinar is available. I would like to thank everyone who took the time to join us today, and particularly those of you who shared your questions and comments. On behalf of the scientists, I'd also like to thank our speakers, Dr. James Crow and Dr. Joseph Jardine, as well as our sponsors, Cynobiological, Absolute Antibody, Thermo Fisher Scientific, Twist Bioscience, Agro Biosystems, 10x Genomics, and BPS Bioscience. Thank you, everyone, and goodbye.